Hello, and welcome back to Introduction to Genetics and Evolution. In the previous video, we talked about these principles of selective sweeps and hitchhiking. What happens is, if you have a region of low recombination in particular, when an advantageous variant spreads through the population, it eliminates variation at nearby neutral sites. It reduces pi, which is our measure of nucleotide variation. Now, what I'd like to do in this video is explore how we can see signatures of past natural selection in the genome leveraging this. Now again, recombination can help us detect natural selection. So we're going to use the principles we talked about in the last video to actually identify areas that seem to have experienced natural selection. Now an important principle which I haven't emphasized up until now is that recombination rates are not constant across the genome. There are areas where recombination is very common, and we already discussed in this context crossover hotspots, but there are often broad regions which have many hotspots, and there are broad regions that have almost no hotspots. So recombination is especially often thought to be low near the centromeres. The centromere is the part of the chromosome that holds together the chromatids. So let's imagine that here's a chromosome, that you have your centromere here at one end, and let's say we're looking at different stretches of a million base pairs. So this first stretch of a million base pairs, let's say the recombination rate is 0.1 centimorgans. So that's a very low recombination rate. We go a little bit further out in this stretch of a million base pairs, the recombination rate is 3 centimorgans. Go a little bit further out in this stretch of a million base pairs, the recombination rate is 5 centimorgans. So an important principle I want to stress from this is that you can't directly translate centimorgans to millions of bases or numbers of bases because there's not a one-to-one transition here. There are areas where very low centimorgans are associated with high numbers of bases. There are areas where the number of centimorgans is much higher for the same number of bases. Okay? The linear order stays the same either way, but the recombination rate may change. And again, as I mentioned, the recombination rate is often lower near the centromere. Now we measure recombination rate as centimorgans per megabase. So let me show you an actual picture of human recombination rates. This is looking across chromosome 3 of humans. So you see here near the centromere, recombination rate is low, but here on the x-axis is position in millions of bases. On the y-axis is the average recombination rate. We see here near the end, the recombination is pretty high, goes down, up, down, etc., but low again near the center. So it's very variable. Now what we can do is we can focus at what's going to happen if you have an adaptive variant in, say, a region like this, where recombination rate is very low, versus if you have an adaptive variant in a region like this, where recombination rate is moderately high. So let's look at that. Again, these new adaptive alleles can arise by mutation every now and then. And sometimes they arise in a region where recombination is low. So again, there's low centimorgans per megabase, no hotspots. So in this case, now we have 5 variable sites depicted here on this figure. They're in blue. I apologize if it's a little bit hard to see on the slide, but I wanted to show you an actual stretch of sequence there. So let's see what happens. Let's put this now in the case where there's no recombination. So here's our new adaptive variant. This is this beneficial G. If there's no recombination, what's going to happen? Well, these stars indicate the variable sites. So there's 1, 2, 3, 4, 5 different variable sites aside from the selected one. So these are ones we're assuming are neutral, that it does not matter for fitness if you have a T or a G here, or if you have a C or a T there. So as this G spreads, you lose variation at all other nucleotide sites. So now none of these are variable anymore. That's why I got rid of the stars. So you lost all variation across this entire stretch. Pi across this whole sequence now, or across this whole stretch, is 0 in this population. Now let's try this again in a region where recombination is very common, where you might have high centimorgans per megabase. Well, here's that adaptive variant again. We still have 5 other variable sites with neutral variation. Well, as it spreads in this case, you see we've already in this case had some recombination out near the ends, right? Well, as it continues to spread, we've lost variation very close to the selected site, right? That maybe there was no recombination between these two, but on average, since there's a lot of recombination, you retained variation out here, you retained it here, here, and here. So again, you maintain a high pi across most of the sequence, but you lost a tiny bit of pi because sequence is very close to the spreading site lost variation. So we can generate a prediction. Our prediction is if you survey regions around the genome, then you should be able to find some sort of association between recombination rate and pi. So take this quiz question real quick. Do you think this relationship will be absent? Do you think there's really no relationship? Do you think regions of high recombination should generally have high pi? This would be a positive association. Or do you expect a negative relationship? The regions of high recombination should have low pi. Well, that was an extremely easy one. Again, as I said, if you have high recombination, you're able to sustain variation. So we do expect this to be a positive association. And in fact, that's exactly what you see. So this is looking at data from humans. On the x-axis here is recombination rate. On the y-axis here is pi. This is from a study by Hellman et al. In Genome Research. And again, you see there's a general trend that as recombination goes up, pi goes up. Each individual circle here is a stretch of sequence in the human genome. So there you go. Well, this is true in humans, but is it just humans? No! Here it is in Drosophila. This was actually a classic study by Begun and Quadro in 1992 in Nature. This paper at the time of recording was already cited well over 550 times. It's a very popular study, but again, you see here on the x-axis is coefficient of exchange, which is essentially recombination. And on the y-axis here is pi, and again, you see this general positive trend. This, again, potentially suggests the spread of adaptive variants. But we'll come back to this actually in the next video. Here's the same thing in various tomato species. That again, you see this higher pi in regions of high recombination. This wasn't as compelling, but it was weakly statistically significant, so that's cool. Yeast, in contrast, I don't actually show you a picture of it, but it actually doesn't really match the expectation. There was no significant positive or negative relationship. And it could be in this case that yeast tend to reproduce asexually very often rather than sexually, potentially reducing this opportunity. Now, let me ask, what does this overall pattern, not the yeast part, but for humans, tomatoes, Drosophila, what does this mean? Well, we see that there's a positive association between recombination rate and pi in many, many different species. Now, this relationship, again, is predicted from the action of natural selection, and not just any natural selection, but specifically the spread of adaptive variants, right? That we have this adaptive variant spreading, and we have a selective sweep associated with it and hitchhiking of nearby alleles. What this means, the fact that we see this positive association looking across the whole genome, is that selection's impact on genomes is huge. That basically selection is happening all the time. We have adaptive variants spreading within species all the time, and that's creating this overall pattern. Selection is strong. Darwin was right. Well, let me ask you, can we leverage it? Well, the answer is yes, that we can leverage this in an interesting way. Now, we expect the regions of low recombination to have low variation or low pi. Now, what would happen? This is actually a fictitious data point. I actually just drew it in here. What would happen if you see a very low pi in a very small region of high recombination? What does that actually mean? So let's say you have this, this region of very high recombination but has very low nucleotide diversity. What might you expect to happen there? Well, the answer could be, now there's a lot of possibilities. The answer could be, it could suggest there was a very recent selective sweep. Could suggest that basically very recently you had an adaptive variant within the last couple of hundred or couple of thousand years spread through all the populations of the species and eliminate variation at nearby neutral sites. Now, what people do and have been doing and are continuing to do is we're using this to actually find genes that have experienced recent selection. Looking across the genome at regions that we know have high recombination and thus should have high pi, high nucleotide diversity. But when we see a small stretch of it that has unusually low pi, that could suggest that there has been a recent selective sweep and that there might be a very interesting gene to study there. Now, this overall pattern I've described to you in the context of this sort of adaptive variance spreading through the population, these frequent sweeps through the population. There is an alternative hypothesis. This alternative hypothesis for why pi and recombination rate may be correlated is referred to as background selection. We'll come back to background selection in the next video.